after the centrifugation, in the solution part we will have our plasmid DNA, whereas the genomic DNA and other protein and contaminants will be precipitated down. So, we will take that supernatant where the plasmid DNA is present, and then we will load that supernatant into a spin column. So, spin column basically have a silica column. So, this is the tube which is the wash tube. So, whatever solution you put here, we need to centrifuge it and the solution will come down to this wash tube, and this column is basically a silica column. When we will load our sample, the DNA will get attached to the silica membrane, and later part we will isolate that DNA using a low ionic buffer. So, now that we have understand the basic steps or the theory of plasmid extraction, we will go and start isolating the plasmid. So, till now I have the cell pellet here. Now, I will resuspend the cell pellet into the resuspension buffer which has trace EDTA and RNase A, and that particular buffer is kept in an ice box because it is written that it has to be stored at 2 to 8 degrees centigrade. Now, all I have is the cell pellet from a 5 ml to 6 ml culture. So, this culture or the cell pellet has to be resuspended into the resuspension buffer. The resuspension buffer contains, as I mentioned earlier, trace EDTA and RNase A. So, for that reason, the bottle itself says that it has to be stored at 2 to 8 degrees centigrade temperature. So, we usually keep it at 4 degree fridge. Here I have taken it out and kept it in a ice box. So, as this has RNase A, an enzyme which is very happy at a low temperature, that is why we need to keep it at a low temperature or fridge. So, now the protocol mentioned here is that I have to take 250 ml of resuspension buffer to dissolve that 5 ml pellet or pellet from a 5 ml culture. So, I will take a tip, I will adjust the micropipette. So, as the protocol mentioned here is I have to add 250 microliter of desuspension buffer. So, first I will take a micro tip, then adjust the pipette man to 250 microliter. So, now it is 250 microliter. Then I will take the P1 buffer. Carefully I will take 250 microliter of this buffer, which is basically the resuspension buffer. Then I will add this resuspension buffer into my pellet. So, whatever pellet I have got from this 5 ml of culture, I will resuspend it. So, resuspension means you can vortex it or you can pipette it up and down. Do not vigorously pipette it up and down, otherwise you might lose cell pellets and also damage the cell pellet. So, here what I am doing is I am continuously taking some solution and again putting that solution back, and by that I am actually resuspending the pellet into this buffer. So, you should not be able to see any kind of pellet or any kind of clump after you are done with the resuspension. So, gently pipette it up and down multiple times so that there is no clump of cell pellet and everything comes into the same buffer. Now that I have resuspended my pellet, I will put this pellet into a lysis buffer. So, again the protocol says I have to add 250 microliter of lysis buffer. Now, after adding the lysis buffer, it is tricky because I need to have a, after resuspending the pellet into this resuspension buffer, I will add the lysis buffer. Now, I have to add the lysis buffer again 250 microliter. Now, after adding the lysis buffer, we have to invert the tube to mix the lysis buffer properly. Now, at this stage do not vortex or agitate too much the cell pellet, otherwise the lysis will be hampered and the DNA, you might cause the genomic DNA to break into small parts. If the genomic DNA breaks into small part, then you will get genomic DNA contamination into your plasmid DNA, because the plasmid DNA is also small, and if the genomic DNA becomes smaller, then it might get stuck to the membrane and you will get genomic DNA contamination. So, be very gentle, and while to mix the tube, the lysis buffer into this solution, you just have to invert it like this. So, I have not added the lysis buffer, because after adding the lysis buffer there is an incubation time and it should not go beyond 5 minutes. So, we will here in our lab we stick to 4-minute time, because we do not want to overdo the lysis process. So, we will stick for 4 minutes, and at 2-minute interval we will mix it by inverting the tube. So, inverting the tube is like this. So, just invert it like this, do not vortex it, do not centrifuge it, do not agitate it too much, otherwise you might cause shearing of the genomic DNA, which is undesirable. So, now I will add the lysis buffer. So, lysis buffer contains high alkaline solution, which is sodium hydroxide, and also SDS or sodium dodecyl sulphate. So, these two will cause lysis of the cell. So, I have taken 250 microliter of lysis buffer, then I will add directly to the tube and immediately I will invert this 2 to 3 times. Now, I will wait another 2 minutes. So, keep a clock beside you so that you can monitor the time, and after 2 minutes I will again mix it for another 2 times by inverting the tube, and then when 4 minutes is over I will immediately add the neutralization buffer. So, at this time keep an eye, and it should not be agitated too much, otherwise it might hamper the lysis process. So, you have to wait another 2 minutes. So, as I have mentioned, the lysis buffer contains SDS, it will break down all the cell membrane because it is an anionic detergent, and it also has alkaline or alkali basically. So, a high amount of sodium hydroxide, it will denature all the proteins and the DNA, RNA, whatever is present. And when we will give the neutralization buffer, the plasmid DNA, because of the size difference and the shape of the plasmid, it will readily anneal and come into the solution, while others will precipitate out. So, here the timing is really important, do not overdo this, and even in the kit itself, the protocol, it is mentioned that you should not keep the solution in lysis buffer more than 5 minutes. Since 2 minutes is over, I will again invert it another 3 times for a proper mixing of the lysis buffer, and again keep it for another 2 minutes. So, a total of 4-minute incubation will be given. So, you can make all this solution in your lab using a molecular biology book, protocol book. So, basically the components, if you know, then you can make it, and if you, depending on the nature or how much plasmid you want to make, you can scale it up. Here I am taking 5 ml culture to isolate the plasmid. You can take out amount 50 ml to even 250 ml of culture. Almost 3 minutes is over. Now, as the next step requires me to add 350 ml microliter of neutralization buffer, I will set my pipette man to 350 microliter, and I will be prepared. Whenever the 4 minutes is over, I will add the 350 microliter of neutralization buffer. So, the neutralization buffer will actually neutralize the solution. Since it was alkaline, it has acidic solution, which is potassium acetate, and just after I give the potassium acetate, I will show you there will be some precipitation and the whole solution will curdle up. So, always keep an eye at the timer, do not overdo this lysis process because it is critical at this stage. Since my 4 minute is done, now I will add 350 microliter of neutralization buffer to this solution, and immediately after adding I will vigorously shake it the centrifuge tube, and you can see the whole solution becomes turbid and small white curd like precipitation will occur. As I have mentioned that the neutralization buffer has potassium acetate. This will form the potassium dodecyl sulfate, which is a salt and it is insoluble in the solution. So, it will precipitate out of the solution, and also the genomic DNA will precipitate out because it is of huge size and it cannot readily anneal with the another strand of its own. So, it will clump together and precipitate out of the solution, and all the proteins will come out of the solution, only the plasmid DNA will remain in the solution. Now, as you can see, the solution becomes too much of turbid. If you feel like there is not turbidity is less or it is not properly mixed, you just do vigorous mixing, and then we will put it for centrifugation. Now, at this stage the centrifugation is for longer time. We have to do centrifugation for around 10 minutes at the highest speed. So, I am putting this into the centrifugation machine. I have also taken the balance. I close down the lid, close the whole thing. I will select the speed of the centrifuge at the highest. Also, I will select the timer and make it at 10 minutes because the protocol requires me to do the centrifugation for 10 minutes. And then I will start. So, when that particular centrifugation process is happening, we have to prepare this spin column. Basically, the spin column, as I have mentioned before, has a silica membrane or silica column. After the centrifugation is over, so we need to take out the supernatant which will contain the plasmid DNA and put the content in this spin column. So, spin column, as I have mentioned earlier, has this silica membrane where our DNA will get stuck, and all other contaminants, like if there is any kind of protein contaminant or any salt, it will elute it out. So, after this step we need to have some subsequent centrifugation step, and here our centrifugation is over. The volume it can hold in this spin column is around 800 microliter. So, I will set up the pipette to 800 microliter. I will take out my sample. As you can see, the supernatant has become clear now, and all the precipitates are all the insoluble fragments have been precipitated out. That contains our genomic DNA, other proteins and salts of potassium dodecyl sulfate. Now, I will take out this solution and put in this spin column. Now, I will put for another round of centrifugation. Put on the lid. This centrifugations are short spin. So, basically I will reduce the time to 1 minute. After 1 minute, we will see that all the flow through has gone down to the wash collector, and we will discard that wash collector. After the centrifugation is done, then we need to have two washing step. One is with a high ionic strength buffer that will provide us to have a clean without any protein or salt contaminant DNA, and after that we will wash it with 70% ethanol. So, ethanol will allow the DNA to be bound at the silica column, but other reagents or other salts will wash away from the DNA, and then we will have a elution step. So, elution will happen at a low ionic strength buffer, or if you do not want to have it in buffer, you can do in a normal autoclaved or non deionized water as well. Make sure the water is nucleus free, otherwise it might degrade the DNA. The centrifugation is over. As you can see, the flow through has come down into this wash tube. I will discard this wash tube. Now, according to the protocol, I have to add around 500 microliter of this PB buffer, which is another binding buffer. So, basically it will ensure that the DNA becomes bound to the silica column, while other contaminants will wash away. So, I am adding 500 microliter of this PB buffer or binding buffer. Then I will again go for another round of centrifugation for 1 minute. After this binding process, we will wash the column using this washing buffer, which contains alcohol, which is ethanol or 70% alcohol. So, I have to add 700 microliter of this washing buffer. So, I have set up my micropipette. So, if you buy a kit, you will see that some of the reagents will be asked you to add some of the stuff, like here before use it is mentioned that add 24 ml of ethanol to obtain 30 ml of buffer PE. So, you have to add those buffers when you get the kit, and then you have to keep it. So, some of the stuff you have to ensure that you add previously before you start using the kit, and those will be mentioned at the bottle of those reagent or even in the protocol card, which will be given by the manufacturer. Our binding has been done. Again, the flow through has been collected in the wash tube. Now, we will wash the column using this wash buffer, which contains around 70% of ethanol. Carefully pipette out this solution, because alcoholic solution tend to drip while pipetting it out. I will again go for another round of centrifugation. Now, here I am isolating plasmid from one particular culture, and that is why I did not label the spin column. If you are isolating plasmid from different culture or different colonies, you should always label all the spin columns, otherwise it might happen that you might mix up with different cultures, and that might cause any problem. After this washing step is done, we need to remove the alcohol. So, I will show you. After I throw away the wash buffer, I will again spin it down without putting any kind of solution. So, that will ensure whatever residual alcohol is present in that column, it will evaporate and it will go away. Otherwise, if a solution contains alcohol, the DNA will not solubilize in whatever buffer you are actually trying to elute out your DNA. So, the column should be alcohol free before you try to elute out your DNA. So, one step of washing is done. So, I will throw away again the washing buffer, and then again I will put this tube at the wash tube, and then the empty tube I will again centrifuge it to remove the residual alcohol that is present. So, all the steps will be written in that protocol kit, protocol card, or else you need to follow a book. After the washing is done, then I will use a elution buffer. So, here the elution buffer is basically a very low ionic strength tris buffer, which has around 5 millimolar to 10 millimolar tris, and it might contain EDTA, which will actually chelate down all the divalent ions, and so that the nucleus, if any kind of nucleus contamination is present, your DNA will be not cleaved, because the EDTA will chelate all the divalent ion and nucleus need those divalent ions to work. So, it might contain EDTA, or else it will have just 5 millimolar to 10 millimolar tris buffer. If you do not want to add this elution buffer, because you might not know the actual components of this buffer, and if there is EDTA present, so you cannot work in subsequent experiment, like you want to restriction digestion of this plasmid. So, there you need to put some enzymes which will again need the divalent ions, and if EDTA is present, then it will chelate all those ions and your restriction enzyme will not work properly. In those cases, you can use just normal 5 millimolar or 10 millimolar tris, pH around 8.2 or 8.4. Otherwise, you can also elute out your sample or plasmid in nucleus free deionized water. So, now that I have removed the alcohol, now I will take out this tube, the spin column. Now that I have removed the alcohol that is present in the wash buffer, I will remove this wash tube because I do not need it anymore. Then I will take this spin column and I will take a clean centrifuge tube or a collection tube. I will put the spin column in that particular collection tube. So, whenever I am putting the elution buffer in this collection in this column, and subsequently I will centrifuge this tube, the collection or elutant will contain my plasmid DNA and will be collected in this centrifuge tube. So, now I will put 50 microliter of this elution buffer. Take one pipette. This is my elution buffer. And just put the buffer at the middle of the tube. Do not touch the spin membrane or the silica membrane basically. Do not make any hole or damage the column, otherwise it might get damaged. And then after putting the elution buffer, you need to wait around 1 minute or so. This time the buffer should be at room temperature. If it is a little bit warm, it is great, because the elution will be perfect at that particular temperature. Do not cool it down and wait for another 1 minute, so that the buffer will spread all over the silica bed, and when you centrifuge it, it will come down to the collection tube and along with the plasmid DNA. Now that we have waited for 1 minute, I will put this whole thing for centrifugation. So, you cannot close the lid of the centrifuge tube, but always close the centrifuge tube, the centrifuge machine's lid, then centrifuge at the highest speed for 1 minute. After the centrifugation is done, you will see that the collection tube contains some solution which is basically the elution buffer. So, basically it has been eluted the plasmid DNA which was attached to the silica bed. Now, you can throw away this spin column, and this is the plasmid you have extracted from the culture. Now, to check the amount of DNA which it contains, you have to check at the 260 nanometer of wavelength, because DNA absorbs at 260 nanometer. There you can also check if other contaminants are present, like other carbohydrates or salt or RNA is present. So, you have to check 260 by 280 nanometer ratio, you have to check 230 by 280 nanometer ratios. So, those ratios you have to check for how much yield you have gained from this extraction process. Now, all the DNA will absorb at 260 nanometer. So, you might not know if all the DNA that are present in this tube are of your choice or plasmid DNA, or genomic DNA contamination is also there or not. Another aspect of plasmid extraction is that the shape of the plasmid. So, the plasmid can be of a circular double stranded shape, and which can be super coiled, or it might have some nick at one strand, or it can be a nick of two strand. So, depending on those nick or your extraction procedure, the plasmid might get distorted and the shape might change. To check if you have high quality plasmid, which is basically the super coiled double stranded plasmid, you need to run an 1% agarose gel to check the plasmid how it looks, and if there is any other contaminant present. If you see smear, that means the DNA has been broken down into smaller fragments. If you see different kind of fragments or at a very big chunk of DNA that might contains your genomic DNA, then you need to check the size and the shape of the plasmid by running in 1% agarose gel. Now, this plasmid, to store this plasmid for regular use, you can store it at 4 degree, but for longer period of time, if you want to store it, you have to keep it at minus 20 degree centigrade temperature. And you can use this plasmid in any kind of subsequent reactions, like you can do PCR reaction, you can digest this plasmid using restriction enzyme, you can use for transformation or transfection, you can go for gene sequencing. If you have put any kind of gene in your vector and you have isolated that particular vector plasmid, you can send it for sequencing to check if the particular gene is present or not. So, based on your experiment, you can isolate how much plasmid you need, how much good quality plasmid you can have. So, based on that, you need to modify your protocol a little bit and try to get as good plasmid as possible. Thank you.